Já třeba jsem teda skopíroval jako do těch ještě e-mailů, takový zrovna.
Jo, takže vysílám. Jo, jo,
Skopíroval jako taky.
jo,
Skopíroval jako taky.
Větší.
15:30.
Tak jenom jestli můžete. Byla mít rozšířenou obrazovku.
No.
Jo, takže tady vlastně to se vysílá.
Jo, a jak to tam bude pouštět, musím prezentaci, když tam nejít. No, už to vypíše.
Ne, to je vlastně tam se streamem, ten první monitor. Že teda tam píše, že jsem může dělat cokoliv.
Takže já prostě to jenom potom vypnu, protože teď už to všechno běží. A já teď nebudu tu kameru nechám tady. Že nějak někdo přijde, nebo to byl nás.
Kansas, yes. Slovo neusnutíme se so microbiology is
Jo, kde máš špatnou prezentaci? To z okna.
Tak to letíme.
Tady jsem přijde. Ale ten propad. Jo, už jako je to takové. Jo. Jo, to někdy.
A teď už teda vysíláme, takže už můžeme odstartovat. Good afternoon, ladies and gentlemen, students, guests, online followers. Let me invite you on half of our student seminar, and the next 1 hour and 30 minutes will be dedicated to the lecture of Professor Charlese Rice from Kansas State University, who is a member of the International Evaluation Board of our university, and because the international evaluation will start this evening, let's say, by a non-formal introduction. So I asked him if he could give us a talk, which I hope will be very interesting. Professor Charlese Rice is an expert in soil microbiology, soil carbon nitrogen cycling, and he is a Nobel Prize laureate as a part of the International Climate Board, correctly, in 2007. So his lecture will be also linked to the role of soil carbon and soil microorganisms in the global carbon cycle and to the climate change and future and all related topics. I hope I did not forget on something, so it's yours. Very much.
All right, thank you. Thank you, Joseph, for inviting me, and it's a pleasure to be on the evaluation panel for your university, but also an opportunity to speak to you about one of my favorite topics on soil microbiome and how it relates to climate, soil health, and soil carbon. So I'm going to present 2 or 3 different experiments to illustrate some principles, but hopefully there'll be enough time for discussion to answer questions and go into a deep dive. So if you got questions, I think we have an hour and a half scheduled, but presentation should be a lot less than that, but I hopefully will stimulate discussion. So feel free to interrupt. I normally used to teaching, and so interruptions and questions are really a good thing. So with that, let's see if I don't trip over this. Okay. All right. Maybe. Will you get closer? Yeah. Was working before. I'll maybe have to get oh, it's advancing there on the screen, but not up here. Right. Yeah.
Yes, but
It's advancing here, but not up on the
it's
screen.
turning here, not on the slide.
Okay, let me see. See what's going on.
That's
This is a long bit.
This is a long way to go.
sales for 3 times. We're not pushing. How is working?
Okay,
Let's try. Yeah.
yeah, okay, good. Okay. All right, so I'm gonna talk, so I'm a soil microbiologist, so I'm gonna emphasize the soil microbiome and how it relates to soils in particular, but to different aspects of the environment. So this report was National Academy of Sciences report. It came out in 2013. It relates to agriculture, but it really relates to soils and the environment, which is, I know, of interest to the faculty and the students here, hopefully. So in the report, they mention that understanding the relevance of the microbiome to agriculture, but I would argue soils in general in the environment, and harnessing this knowledge of the microbiome to improve crop production, transform efficiency for animal livestock, and increase resilience to stress and disease. So the microbiome is critical. The understanding of the microbiome is critical. And I added this second bullet statement that really the relevance of microbiome to soil is important to understand plant health, soil health, remediation, because I know it's of interest to your faculty here, and also to resilience. So that was originally put into the report, and I added to that. So I'm going to describe a couple different experiments to understand how soil management, land management affects the microbiome, and then the carbon cycling in that. So this is an experiment. We just actually got another publication from this this experiment. So it's a rain-fed continuous maze. I would never do an experiment monocropping for 30. This started in 1990, so it's been what 35 years now. We're starting our 36th year. But it was started out with this 3-year experiment. I foolishly kept it going, and now I can't stop it because there's some historical aspects of this experiment that is fairly unique, and I'll explain that. So the experiment was no-till systems, continuous maze, and then pinch till, which was a chisel disc, if you're familiar with agriculture systems. So not extensively disturbed, but moderately disturbed. And then we were looking at nitrogen leaching in the system originally. And so we were preparing organic nitrogen source for the corn, for the maize, and then mineral synthetic fertilizer as a nitrogen source. And then we also have in here a control where no nitrogen has been applied for 35 years, which has been really interesting. It's a mollisol or turnzim in your in a good prairie soil here. So I'm going to talk a little bit about, I'll talk about soil health, but I have in my soil health metrics 3 key parameters. I call it the Holy Trinity of soil health. And if those 3 components are so organic carbon or organic matter as the key point, but if there was only one thing to measure, it'd be carbon. But really, if you allow me to expand to the 3 key components of that trinity, one is microbial activity, and this third one is some measure of soil structure. And I'll talk about what we're doing to measure that. But they're all the 3 of those are interrelated. Carbon, organic matter supplies fuel for the microbes to operate, and that encourages that nutrient cycling and reuse and cycling of the nutrients contained within the organic matter. But then the microbes also, as I'll illustrate, help interact and build soil structure, build aggregates. And then within those aggregates, carbon is sequestered, protected, and so then you get more carbon stored in the soil. But if you have those 3 components, then you have a healthier soil, and hopefully I'll try to illustrate then that soil is going to have better structure. When it has, with climate change, more intense rainstorm events, that water is going to soak into the soil, infiltrate into the soil, and then be retained by that during the dry periods that occur between those episodic rainfall events. But also you have organic matter that's going to supply nutrients for plant growth, and the microbial activity is going to help recycle and make those nutrients available. So I'm going to go through, and first I'm going to talk about the carbon. So the value of this experiment, this was after 34 years. So experiment started in 1990, and we started taking deep measurements in 1992. So we have some original samples of 1990, but not extensively. But in 92, we have samples collected down to 90 centimeters. And you'll see that we've actually archived some of those samples, stored them in a barn, so we can go back and reanalyze some samples. But what you see here is some unique aspects. So we've been applying organic matter or compost to these soils. This is the top 0 to 5, 5 to 15, and 15 to 30. And I'll show you deeper in the profile a little bit later. But what's interesting about this, and we just got another publication from this experiment just a couple weeks ago in the European Journal of Soil Science. But what you see here is that we were applying organic matter, manure, as the nitrogen source. And then this was fresh manure, but then we lost that source and we had to use composted manure, organic material. And with composting, the nitrogen is less available. So we had to apply more material in order to get the equivalent amount of nitrogen available for the maize crop. But what happened was organic carbon was increasing up until the time we switched from fresh manure to composted material. It jumped, increased because we were applying more material. But then interestingly, it flattened off. We're quibbling with the reviewers. They want to call it equilibrium. I'm calling it saturation. But you'll see, so the conventional wisdom would say this soil is done sequestering, can't hold any more carbon. But what happened was, then if you can see here, after it reached equilibrium or saturation, then we had carbon translocated to the second layer, and it started to increase. And so it was increasing for the first 15 years, 17 years or so. But then as this reached saturation, carbon started to move down. This is the no-till system, and has been increasing to current day. But now we're reaching some sort of higher level equilibrium, and even down to 15 to 30 in the no-till system, we're starting to see an increase down at 15 to 30. So it didn't happen initially. And some people argue, well, it was animals or earthworms mixing that material, but it didn't. It would have been happening the whole time the experiment was going on. So we only get carbon movement until when the surface becomes saturated or equilibrium. And so you'll see here in a moment a potential explanation for that. And this is the magenta line, purple line is with the tilled system. Both, because you're mixing it in, it's also carbon is getting mixed into the second layer, but it's not increasing in the 15 to 30 until just the last couple of years. So we've done a calculation, and then just because of the tillage, we're losing 30% of the carbon that we added due to tillage and then respiration by the microbial activity. So that no-till is holding on and sequestering and allowing the carbon to increase in that soil. Okay, so we can only see this because we've kept this experiment going for 30 plus years. That was unique about it. Okay, so if you look at the whole profile, you can see here the no-till has about almost 5.5% organic matter in the surface. We're seeing that increase, or it's higher than 515. and then that slight increase that we're starting to see now at 15 to 30. The no-till with just synthetic fertilizer, we also have seen an increase, but it's only in the top 5 centimeters. We're not seeing it in deeper layers. And what I'll suggest to you later is that no-till is absolutely critical, but if we want to improve carbon down through the profile, we need more higher inputs of plant residue or organic material in order to reach the true potential of that soil to store or sequester carbon. Okay, if we look, Wazama Mushtag just recently analyzed the soil, and what we estimate, we've seen that between 1992 and 23, that the organic fertilizer compost treatment had a net gain of 10% over that time period. The tillage has actually, through the profiles, lost carbon. Even by adding organic material, we're still losing carbon, and I'll explain that. We're losing it down deep in the profile. The synthetic fertilizer, this one here with no-till, we're still losing carbon, mostly because of the deep soil profile. The roots aren't getting deep enough. There's not enough material in there. And then without any nitrogen inputs, the corn, maize is not growing enough, and we're losing anywhere from 6 to 20% of the profile carbon down to 90 centimeters. So it turns out that this experiment, when we started it, was amolosol. It would have been C4 grasses, but about 50 years ago, this farm was planted to mostly small grains. So a C3 plant, wheat, oats, and that. So now when we started this experiment, we had a mostly C3 signature, C13 signature in that profile. And with the corn roots being C4, being more negative, or less negative, sorry, minus 12 C13 content, we could determine how much of the carbon in that profile was coming from corn, and actually we can determine how much was coming from the compost. What I'm showing you here today is the C13 signature from the corn. So it's around minus 12, and we're looking at the difference between 1992 and 2023, how much corn carbon is being put into the system by layer. So we went back and analyzed. The administrators were complaining about all this soil stored up in the barn. We went back and reanalyzed those samples for C13 content. And what you see here, I think, is some interesting results. So 0 to 5 centimeters, well, you can see the corn root carbon, or residue too, that we're getting net gains in carbon coming from the corn. Not surprisingly, even in the control system where we haven't been putting on nitrogen for 30 plus years. So there's a little bit of roots and residue being added to the soil. 5-15, you can see here the no-till, much more greater gains than the till system, but again, we're getting better root growth or more residue. It could be residue and or roots. And so that we get higher production and more carbon put in from the corn. At 15 to 30, now we're starting to see some differences. The till system, where we haven't been putting on nitrogen, we're not getting as much productivity. So now we're seeing less root. I'm interpreting this means less roots getting down 15 to 30 centimeters. We're actually losing carbon in that till system. The no-till, with mineral fertilizer, that we're still getting better production, not much, because of water relations with the no-till systems. And so we get a little bit of root growth down here with the control. But as we go deeper in the profile, it becomes, I think, even more separation between the systems. So at fit 30 to 45 centimeters, the tilt systems, we're actually losing carbon. And there's not suggesting that we're not getting the roots getting down there as much in that tilt system. And I'll explain that here, what I think is going on. In the no-till, we're still getting roots growing down, a little bit of carbon added, not much, but still measurable, even down to 45 to 60 centimeters. And only when we get to 60 to 75 centimeters do we see a net loss of original C4 carbon. Okay, so what's going on? My hypothesis is that there's two things. One is that the no-till systems, because they're not disturbed, you get old root channels being formed, we're getting better porosity, so roots are growing deeper and depositing that root carbon, that modern root carbon down deep. The other thing is that with tillage, and some other people that worked in water relations have shown this, is that tillage, you get a flush of nutrients because of till, but then the roots are staying up near the surface and not growing as well deep because the porosity and the nutrients are keeping the roots nearer to the top 30 centimeters. So actually the no-till systems are allowing roots to grow deeper and depositing more carbon. Even though we've lost carbon overall, we're getting more modern carbon in that system because of the porosity and probably nutrient cycling. Okay. I think it's really neat. Yeah, thanks. I think it's cool. All right. All right, so that's the part of the carbon story, and I'll come back to that. But if we look at the three-legged stool, then the other thing is microbial activity. So again, using the 30+ year experiment, what's going on with the carbon and microbial relations. So again, this was the article that was just published a couple weeks ago. What we've shown is that with the compost added, this is microbial biomass in the top graph measured in 2017. I guess the label got cut off. And then this was measured in 2023. So we're measuring microbial biomass by phospholipid fatty acid content. And what you see here is that, again, this is why I'm calling it saturation, is that organic matter carbon has increased in 17 and further increased in 2023, but we don't see an increase in microbial biomass. That's why I call it saturation. There's so much carbon there that we're adding that the microbes are constrained by weather, the climate conditions, that they can't use up that or process that carbon. That allows that carbon then to translocate to deeper layers. The reviewers don't like it. They're thinking chemical saturation. I'm calling it biological saturation. That's my term. They don't like it. But whether it's equilibrium or saturation, the point here is that we're adding so much carbon to the system that the microbes can't process it. And then that carbon is then translocated to deeper depths. And if we look at further, Azama, she's working on her thesis. Actually, I got it in my computer. I'm supposed to be reading it. But if you look at the carbon concentration and microbial biomass, again, over the top 3 layers of soil, you can see, again, we're reaching some sort of equilibrium or saturation, plotting that. And that, I think, is then why then that carbon, extra carbon that we're adding every year is now being moved down into subsequent layers. So the question is, how much carbon can this soil hold? I don't know. The wisdom would say this soil can't hold any more carbon, right? But after 34 years, we've seen it move from 0 to 5, 5 to 15, and 15 to 30. I probably won't be here in another 34 years, but who knows? It will move down to 30 to 45, 45 to 60. I don't know. All right. So the capacity of the soil to hold on to carbon I think, is unknown. And so, but it's important to do minimally disturbed soil systems, no-till. And I would propose also, you'll see is how do we get carbon deeper into the soil layers quicker? We've also looked at enzyme activity. I'm reviewing her chapter. She's done these polynomial equations, lines. I don't like that. But again, because of the soil enzymatic activity, we can go back to those samples that were archived in 1992 and track how enzymes in air-dried soils have changed over time of this experiment. And so what you see here is, this is beta-glucosidase activity, an enzyme important for carbon cycling. And in the top 5 centimeters, it increased, even though this shows the decrease, ignore that, it really probably increased and then flattened off. In the subsequent layer 5 to 15, it has increased, again, in both no-till and conventional system. And then in the bottom, it really was flat until this, I would argue it's probably a linear plateau, linear here. And then now as we're getting carbon into that 15 to 30, we're seeing beta-glucosidase. We've looked at aryl sulfatase, NAG, N-acetylglucosamidase, and phosphatase. We've looked at 4 different enzymes, and they're showing somewhat different patterns, except for beta-phosphatase. But so we're seeing more microbial biomass, we're seeing more enzyme activity, particularly as those deeper layers receive more carbon. So we're getting more active soil biology in that top 30 centimeters, even without tillage. Most of the microbial biomass, generally, we see an increase across all the microbial groups. Things about phospholipids, you separate out gram-positive, gram-negative, actinomycetes, fungi. One exception of that increase is mycorrhizal fungi. And what you see here is that with the compost added, we're seeing decreased AMF abundance in the soil. Why is that? I'll just put in my teaching cap for the students out there.
Might be related that they are not important in such.
Right, well, we were talking about nitrogen, but in the compost, you're adding a bunch of other nutrients. We're adding phosphorus, and phosphorus is really high in these soils. And so what happens is that mycorrhizal fungi are important. The plants use the mycorrhizal fungi to help uptake phosphorus. Well, if there's plenty of phosphorus in there, why do I need the symbiosis? So we actually see a decreased abundance with the compost added. They're still there, but they're not. The plants are not relying on the mycorrhizae to collect phosphorus. Here in the control, there's no nitrogen added, but the phosphorus is low, and so now there's an increased reliance on the mycorrhizal fungi to scavenge for nutrients. And so we actually see the opposite effect, is that there's a greater demand for that mycorrhizal fungi, for that symbiosis to acquire nutrients, particularly phosphorus. So it was interesting to see these results. And if you do, Zama did a PCA analysis, and you can see here, it's interesting in that the 2 systems that had the compost added, this is the no-till, and then this one here, the magenta, is the tilled system, that they are somewhat aligned. But then if you look at the synthetic fertilizer, this is the no-till system, and the blue is the till system. So we have different responses, and then the control, they're centered around here. So actually, the compost, the tillage is having some effect, but this nutrient nitrogen source is having a dramatic effect on the composition or the PCA analysis for carbon, nitrogen, and enzyme activity. So you can see that we've created some different environments and systems for that after 34 years. Okay, so now we're going to talk a little bit about soil structure. Wrong one. So the other component of what I call the Holy Trinity, carbon, microbiology, and soil structure. So the way we measure soil structure is by what we call macro aggregate, looking at macro aggregate or aggregation, macro aggregate stability, wet aggregate stability, sorry. And so there's different ways to do that. You can do infiltration, you can do other means to measure soil structure, but we've used wet aggregate stability. And so what you see here, this is not up to date, but Mika analyzed aggregates. And what she did was look at aggregates greater than 2,000 microns. We call those the large macro aggregates. And then aggregates greater than 250 microns to 2,000 size. So those are the kind of the still macro aggregates, but the smallest macro aggregate size. But both cases, what you see here is the no-till is the dark, plots. The tilled system is the hatch, and then we had a native prairie, undisturbed native prairie, tallgrass prairies, as a kind of a reference point. And what you see here that over time that we're starting to build, the no-till system is building up those macro aggregates. We're getting closer to the prairie, and we've got some evidence that it's getting fairly close now to 2023. But you see that we're building those, and I'll try to explain why we think that's happening here in a minute. So we've done other aggregate analysis that we're waiting for, but we have an opportunity to work with the Danforth Center that's doing XRT, X-ray tomography on intact soil cores. So these are 5 centimeter cores down. I believe this one is from 15 centimeters. And so we take intact cores and they do analysis of X-ray on these cores. You can see them sitting here on the instrument. So they can look at the entire core, they can reconstruct that core, look at it in 3-dimensional way. And so what they've done, I've taken all the detail that I don't understand on this, but what you can see, what you should see here, this is the native prairie undisturbed for thousands of years. How much hydraulic conductivity or really poor continuity is occurring in those soil cores. This is the no-till with the compost added. And so you can see here there's some variability, but it's actually pretty similar to what the native prairie is. So now after 34 years, we're building more continuous pores. That's the reason why if you go out there after a rainstorm event, the next day, you can walk on it, you probably could plant on it. The tilled system doesn't have as much porosity. There's water still standing in it, on it, or in it, and doesn't have that porosity. So now we're building that more porous structure. Why is that important? Because with climate change, what's happening in our area of the country is that we're getting an increased number of what we call extreme events. So we get, 5 centimeter, 8 centimeter, 10 centimeter events in a few hours. And so we're getting these intense rainstorm events. If we got this soil here, what's going to happen? It's going to wash off and take the soil with it and erode. So, and further soil degradation. The no-till system can absorb that water. And when it's dry between the times, it's holding on to that water and allows those plants to grow in between those, or during that dry period, in between those extreme events. So for the climate change aspect, this soil here, the no-till system, is going to be much more resilient and adapt to climate change than these soils here. So now we've created a system, an ag system, that's more adapted to and help mitigate or adapt to changing climate. So that's why this is important. Okay, so that's that 34-year experiment. I'm going to switch here. Another experiment that shows some of the principles of the soil microbiome related to soil health and carbon. We're blasting Kansas for several reasons. One is we have native prairie. This is a tallgrass prairie that runs across the state. It, you saw on the title slide, it has deep roots, a diverse plant system. Where's my botanist there? So it has Atropogon, Girardii, Sargassum nutans. I've got the other keystone species at the moment, but they're really, really critical. But if you look at across the state here, we also have ag systems across the state. They're going to have shorter root systems. This is an annual wheatgrass. This is, perennial, and then what you barely see is the roots from a annual wheat system. So the roots don't grow as deep and aren't as fibrous. And so the prairie soils have over thousands of years developed from these deep root systems, and now we're tilling them, hopefully no till, but the root system is not as deep as the perennial system. The other thing what's unique for Kansas is that we go from, we were just talking at lunch, we go from 420 millimeters of precipitation a year to over 1,030 millimeters of precipitation a year. There's more precipitation gradient across the state of Kansas than there is from Kansas to the east coast of the United States. So if you want to understand climate and precipitation, Kansas is a perfect living laboratory because we have that climatic gradient across the state. So what we did was, let me go back here. So what we did was we have several sites, and we actually ended up with 12 sites, this precipitation gradient, sampling ag fields, native prairie, and in some cases we had restored prairie, restored, either abandoned and went back to natural or purposely planted prairie. And so in that, what we did across this precipitation gradient, we sampled down to, we actually sampled down to 1.5 meters, 2 meters in some cases for these. But what I'm showing you is the top 30 centimeters or so in here in the topsoil. And actually, we did one of these graphs. I think I showed the subsoil. But what you see here is the left panel is carbon in the native system, in the native prairie, and then the ag systems, the corresponding ag component. And then we looked here for the microbial biomass in the native and the ag. What was interesting here is, as you might expect, more precipitation, more carbon in the soil, right? Because you got more plant growth, more roots, and we get it deeper and more carbon input into the soil. It's less so with the ag system, but the relationship still holds. When we start looking at some of the fractions of microbial biomass, same thing, more carbon, more precipitation, we get more microbial biomass. But what happened in the ag? Actually, there was decoupling. By that disturbance, it's actually slightly negative, whether that's significant or not, but basically flat line across. So now by tilling that soil, and these were all tilled samples or ag fields, that we've decoupled the relationship between precipitation and carbon/microbial biomass. So that active tillage has affected that relationship. And if we go back and look at a few of the sites, the mycorrhizal microbiome and fungi. We had 3 sites going across the middle of Kansas, so this would be 600 millimeters precipitation, 800 to almost 1,000 here. So more microbiome. Here we're getting more in the native prairie. The ag sites, we don't get that. We can see it's greatly reduced. And if you look at the mycorrhizal fungi, the same thing is that mycorrhizal fungi is greatly reduced because of tillage. Because you're breaking open or breaking up that hyphal network that extends out in the soil by tilling every year. And so we lose the mycorrhizal fungi. And in fact, and then if you look at the aggregates, because what I'll show you is that there's a relationship between the amount of fungi, particularly mycorrhizal fungi in the soil, and aggregate structure. And you can see here that I guess I got it reversed, but the ag has less aggregate structure than in the native prairie. And in fact, when we combine all the sites together, what you see here is a relationship of mycorrhizal fungi and aggregate structure. You get somewhat, a significant relationship, except for here's all the ag sites. Lower mycorrhizae, no aggregate structure, or very little. So that tillage, that disturbance is really affecting that hyphal network. So again, I'm a big fan of less disturbance no-till systems, and particularly species that will support the mycorrhizal fungi in that help build that soil structure to make that soil more resilient to changing climates. Okay, and to further show that relationship, hopefully most of you have seen this infamous diagram. Julie Jastrow drew this diagram, and I think I've seen it around the world. But what you see here is this diagram showing the relationship between building up macro aggregates here, but it's that aggregate structure is held together physically and chemically, glues that are produced by microorganisms, particularly the fungi that help physically bind those aggregates together to build that soil structure. And so, and you can actually see these visually. I think this is really a cool photographs. I wish I could claim these, but Mike Miller and Julie had these diagrams, and you can see here the hyphae helping to bind these soil particles together. And can you imagine just going in and disturbing that hyphal network? Now you're just opening up those aggregates that are holding that soil together and carbon inside those. In fact, what we've done, hopefully you can see that, is one of my students, Gail Wilson, showed the relationship amount of hyphae extending in the soil and aggregate structure. And you can see here, there's a really, really strong relationship between amount of hyphae and the amount of macro aggregates there. And if you look at the carbon that's stored in the soil inside those aggregates, again, there's a 3-part relationship: more hyphae, more aggregates, more carbon, because that carbon is entrained inside those aggregates. And again, this is a great demonstration if you want to show students or the public. You take a soil that, I was going to do that. Take it, even a tilled system that has less aggregate. The aggregates are there, but they're not strongly held. You wet it up, and like I was talking about earlier, if they dissolve and then they go downstream. Okay, but in a no-till or a prairie system, the aggregates are more tightly bound together because of the hyphae, and so when you wet them up, they still maintain their soil structure. Okay, so that's the surface. I showed a little bit about the subsoil. So how can we affect the subsoil? So I'm really interested in this. This is, again, some of Zama's work in the long-term experiment that I showed. You can see this is mycorrhizal fungi. You can see, I show you the data from the top, I think it was 30 centimeters or so. In that previous, if you go down through the profile, you can see there's very little mycorrhizal fungi, all the way down to 90 centimeters, regards to tillage. So again, that organic material, high phosphorus, is affecting, even though the phosphorus is still at the surface, is affecting that mycorrhizal relationship all the way deeper in the profile. The control and the mineral fertilizer, you can see again that the no-till systems, because we're not disturbing that network, we're maintaining more mycorrhizal fungi in the soil, and so it's higher, particularly when you don't till the system and you don't load it up with phosphorus. Gram positive, this is similar. You look at gram negative, gram positive, actinomycetes, the relationship is pretty similar. The no-till is generally higher, and with the compost material, again, because we're moving that carbon down, we're building up the potential for more microbial activity and more microbial biomass. And you can see here that's elevated down at 15 to 30 centimeters. Particularly the compost, not so much in the non-compost synthetic fertilizer, we're affecting it down to 15 centimeters. And then there's very little change in composition of microbial community at depth. So how can we stimulate deeper carbon or affect deeper in the profile below 15 or even 30 centimeters? So one is looking at deep-rooted plants. I know some of you are working with miscanthus, and we had a project that we're looking at miscanthus as a potential biofuel crop. But also, this is switchgrass shown here. And so we had where we were looking at root growth. This is soil carbon over time. And you can see that we mapped out the 2009 up to, so 10 years or 8 years later, we're getting carbon put down deep. But even in like sweet sorghum, oh no, I'm sorry, not sweet sorghum. Yeah, sweet sorghum. These are annual crops, potential biofuel crops, but we're getting carbon put at depth in those systems. So that's another way, instead of adding compost or organic manure at the surface, maybe we can grow our carbon down deep. And so we need to go look at our C13 signatures in those soils to track that carbon. That's another way to do that. I was telling at lunch, I also started working in, well, I've been working in Brazil, but I'm working at a project in Mato Grosso where they're doing an integrated crop livestock regenerative ag farm. And so they do a system where they are looking at soybean. So it's a temperate, or a subtropical environment. So they grow soybeans. They harvest the soybeans, plant no-till corn right into that, right after that. They harvest, and also they interseed brachiaria, which is a tropical grass in there. And then they harvest the corn for silage. And then the grass, the brachiaria comes up, and then they either hay or pasture that grass for the cattle. It's a small 70,000 hectare farm with about 70,000 head of cattle. It's like crazy. But what I'm showing you here is the brachiaria after 2 months of growth. 2 months of root growth. And then what happens is at the end of the day, August and September is their dry season. They'll go in and kill the grass, no-till it, and plant soybeans. So this is every year there's root carbon input. This is down, I'm 6 feet, so I'm what, not quite 2 meters tall. This is actually down to 2.5 meters, 2.5 meters of root growth. That's a crazy, I couldn't, they dug the pit for me because they knew I loved deep soil profiles and carbon roots, but this was, I just couldn't believe it. And so we're going in, and this is fresh data. So what we've done is they dug 17 soil pits for us on the farm, and they dug them for us, thankfully. And then we measured carbon. I should have taken the picture of us in the soil pit, but this is the amount of carbon in the top 30 centimeters and then down to 210 centimeters. So this is the integrated crop livestock system, the soybean, corn, pasture grass back area. This is the control is brachiaria, just year-round pasture, and then these are with supplements, and then a rotational graze system. So what you see here in the top 30 centimeters, yeah, there's a little bit of difference there, but if you measure down to 210 centimeters, 60%, 66% of the carbon is below 30 centimeters. They're only measuring the top 30. So how much carbon is there being put down in that system? So what you can see here, there's a 20 significant increase, about 20 megagrams of carbon, and most of that is going to be down below 30 centimeters. And it's the same, similar amount to what the undisturbed native forest is. So we're looking at now doing a carbon balance. And I was just had a phone call this morning with a guy that's supporting our research, and he asked if this farm is carbon neutral or carbon negative. I think that it's carbon negative, because we've gained this amount of carbon over 10 years, and we're doing an LCA, life cycle assessment, on this system. But because of the synergy between the soybeans and the corn, he's not applying as much nitrogen, so his N2O emissions are going to be lower. So his life cycle assessment is at the life cycle assessment of the integrated crop system is significantly lower. And if you add the carbon that they're not generally accounting for below 30 centimeters, I think we're going to be carbon neutral and likely carbon negative for that system. Okay, really interesting, unique opportunity. And the nice thing is that in this integrated crop livestock, he inherited this farm from his grandfather, and he's been instituting regenerative ag practices over a 10-year sequence. So he has fields that are 10 years old, 7 years old, 5 years old, and 3 years old. So instead of waiting 10 years, like I did wait for 30 years, he's got a sequence right now. So we got a temporal sequence that we can measure the changes in soil health and carbon over that time. So that's what we're doing right now. Okay, so this shows that by doing the integrated crop livestock system, that we're gaining about 20 megagrams of carbon over that 10-year period in that. And so we'll see when we get the life cycle assessment in that, but I'm betting that it's going to be close to neutral, if not negative. Okay, so to summarize, I'll try to leave time for discussion here. So one is that I think that soils can reach biological saturation. For anybody to review these papers, well, we'll call it equilibrium, but I argue it's biologically saturated. And then what's unique about this, like running these long-term experiments, is now that we've reached a leveling off in the surface, and a lot of people say that soil can't sequester more carbon. But we're showing that carbon now can be moved down to underlying layers. It fills up and it moves down. So that's the question, is how much carbon can the soil hold? I don't know. And with that equilibrium, then it looks like the microbial activity doesn't increase beyond that. There's more carbon there than the microbes can hold. That allows for the translocation. We are seeing changes in microbial composition. I didn't show the fungal data, but there's an enhanced level of fungi. Again, and AMF in some of those cases, and that helps to support that aggregation. Even if the AMF is suppressed, that hyphal network is there, in particular with the fungi, I think we're still bidding up those aggregates. So in my mind, when I look at phospholipids, microbiome, I'm targeting fungi as an indicator. We're seeing changes in physical structure. This soil is evolving. We're seeing not only carbon, microbial, now we're seeing structural changes, greater pore continuity, which is going to be really critical for our changing climate. That soil is going to make it more resilient. And we're getting, I think we're close to what the native prairie is. But then really the challenge in these non-perennial systems is how do we get carbon down deep faster? We can add compost to the surface, but it takes time. Can we grow that carbon down deep? And the perennial roots, putting perennials into rotation. I know there are some land managers, they grow annual crops, but then they might break that sequence and put it into a grass for 3 to 5 years to rest that soil. But actually, I think they're building soil structure and carbon down deep and putting it in a rotation scheme. I think that's going to be really important, particularly as with changing climate, that we're going to need soils that are going to be more resilient and healthier. So with that, I'd like to thank over the years, this probably doesn't include all the funders in these projects. And okay, I had a slide there that showed any questions, but I guess it disappeared. All right, questions? You didn't interrupt me enough. Yes.
So, yeah, let's start. I'm a chemist, so
Okay.
bear with me. Yeah, I like the carbon story, and I just wonder, it's kind of a static story from my perspective. Do you know something about, or do you care about the turnover? Whether the new compost that you add, whether how much of the carbon, a year later is there from the last year or the year before, or this kind of.
Kind of. So, well, we know that tillage, we're losing 30%, 35% of the carbon that we've added. Because if you look at how we're accumulated over time, that just the act of tillage is stimulating microbial activity. It's also breaking up aggregates, doesn't affect the storage capacity. But we also know that the reason why I focused on fungi, and that's one of the next projects I need to work on, is we know fungi, that their carbon, they cycle carbon more slowly, they're more efficient with it. Bacteria are wasteful, right? They respire more of their carbon that they assimilate than they hold. And so the fungi, the cell wall material, well, they're slower growing, they're more efficient. They're considered K strategists, if that means anything from a microbiologist point of view, but they're more efficient, so they're slower growing and they retain more carbon in their cell bodies. But also the other thing is that their cell walls are more complex. And so the cell material doesn't completely degrade as fast as bacterial cell walls. Okay, they have chitin, they have other components in their cell structure that makes the leftover material when they die, and necromass, is more of it's retained in the soil. So I don't know if that completely answers your question, but people are looking at carbon use efficiency. And we know that fungi are more efficient and they retain more of the carbon, and then more of it is retained in the soil system. But then there's also this protective mechanism, physical protective mechanism, where the hyphae retain those aggregates, and there's carbon inside those aggregates that are physically protected, not chemically protected. So that's what happens with tillage. When I teach, I have a plow running through an aggregate, and it breaks open, and then the microbes, it's like going into a candy store. They get a sugar high, right? And they just, if you have kids, they bounce off the walls, right? And so the microbes are just going in there, eating up all that sugar, that carbon, and respiring it. And so that's the other reason. So there's a chemical and a physical nature of that protection mechanism.
Thank you.
But if anybody works in organic matter, organic carbon, there's a myriad of carbon compounds, and it's a mess, right? A chemist, there's not a definition of organic matter. There used to be the soil chemists used to do these extractions called humic and fulvic acid, and that's if you read Johann Lehmann's work, that idea of soil chemistry doesn't hold true for modern organic matter. No offense to the chemists.
Thank you.
Yeah, just
I have here one question from the chat.
Okay.
Online. That's more up to 20 online people, which is good. So apart from no-till, organic fertilizer and deep rooting crops, what other practices would you recommend to enhance carbon sequestration?
so minimal disturbance is the key, right? First, that no-till is the ultimate, right? But if you could do minimal disturbance, so things like perennials in the rotation, but no-till systems, even with annual crops, you get some gain in carbon. So that's absolutely critical. Start tilling soil, you lose 30% what you get, what you put into the system automatically. And so, yeah, I had a slide in some of my presentation about future innovation. So one is, we talk about deep roots, but then I don't know how popular it is here, but in an annual cropping system, like corn or soybean, maize or soybean, there's always this period where there's nothing growing. And so we're in the US and around in Brazil or in South America, they're doing cover crops. So they plant a crop right after the main cash crop, grain crop, to keep that's the principle of regenerative ag, is keep the soil covered, keep living roots year round. So you don't have to have a perennial, but if you have an annual crop, which if we're going to grow corn, soybean, wheat for food, right? Then you plant a cover crop right after that to keep living roots in that system. So as much roots as you can cram into that soil is really important. I'm challenging the plant breeders. Any plant breeders probably not in this faculty group, but I'm challenging the plant breeders. What do the plant breeders think of when they're trying to breed for wheat, corn, or soybeans? Yield. We need to get their heads in the ground. No pun intended, maybe pun intended, but we need to think about what's going below ground. I would love for them to think about breeding for root traits. Are there root traits, one that would affect the chemistry of that root to make it harder to degrade, so that it resides longer and then it slowly degrades? Or, because I'm a soil microbiologist, can we look at the rhizosphere ecology? Can we have root traits that would selectively recruit microbes that would partner either like with mycorrhizal fungi to help nutrient uptake, with building aggregates, can we have roots that would select for certain microbial communities that would affect carbon and nutrient cycling? We actually just got a position, faculty position advertised in synthetic microbial communities to look at, work with the breeders to see if we can look at synthetic microbes, whether it's bioinoculants or roots that could recruit certain microbes for certain traits, like for aggregation or nutrient cycling. So those are a couple innovations. The power of that first statement, microbiome. I have a slide that we only know 1% of the microbes in soil. Or, well, I'm sorry, we can only cultivate 1%. What are the other 99% doing? We know some genes, but we don't know what they're doing. There are a lot of microbe DNA is unknown. If you do a DNA analysis, they got all these unknowns and we try to match them to some knowns, but we don't know what they're all doing in the soil. So I think from microbial biomass or microbial ecologist point of view, the world is wide open. We got this huge opportunity to look at the soil microbiome and connect it with the plant system. Yeah.
What's your opinion about how these findings may shed different light on the problematics on loss of permafrost due to global warming?
Yeah.
Does it change the picture, or could we somehow use it to combat the effect of greenhouse effects?
Yeah. So, yeah, we really don't know what happens to the permafrost with climate change and warming of the permafrost. There's a lot of experiments out there. I'm somewhat critical of them because they warm the soil and they see more microbial activity, and then you get more carbon dioxide or more methane if they stay wet, and methane would be 30 times more potent than CO2, so it has a feedback effect. But that's not the whole picture with climate change. They're only looking at the temperature effect. But if you have warmer temperatures, what happens to water relations? More evaporation. So they're assuming that the water's going to be staying, they're going warming up, say, have ice in now water. Well, you might have more evaporation. So instead of saying wet, they actually might get drier. And then you would have less microbial activity because water is the constraint there. But I don't know. Now, I mean, sorry for being critical, but there are a lot of these experiments that are done in the lab or whatever, they just warm up the temperature, but they don't look at the water relations. So whether that, definitely you're going to have warmer temperature and more microbial activity, but then eventually, depending on water relations, you actually might have less microbial activity. So, we don't know the whole picture. It's still an unknown, but it is kind of scary because all that carbon that's stored, because there's a massive amount of carbon stored in those frozen soils, and if they do warm up and they stay wet, and they go to methane. Now, if they don't saturate and they go to CO2, then the feedback, I mean, it's still an effect on the climate, but not as much as if it's methane. But they talk about this giant methane bubble, or that could increase with melting of the permafrost. That happens, then, everybody wants to hold temperatures to 2 degrees centigrade, which we're almost there now. Yeah, Katie, and it's kind of scary then what would happen, because there's a massive amount of carbon in permafrost soils. Does that answer your question?
Yeah.
Yeah.
Another question from the chat. Did you do any trials using biochar?
Okay. Yeah. Yeah, we haven't done any biochar. I'll be open. I'm not a big biochar fan. I mean, it is a recalcitrant carbon that you can add to soil. But living in the Midwest, I mean, in the mollisols, and you have them here, right? Think of how much biochar you would have to add to a good prairie soil mollisol in order to make a difference. There's a massive amount of carbon there. Think how many truckloads, how much energy you would have to take to transport that biochar and put it on thousands of hectares to really make a difference. Now, the opportunity for biochar is really on, we're talking lunch surface mine soils, reclamation. Soils that are highly degraded. That if you want to restore some of those old surface mine soils, they're very low in carbon, okay? Or other soils that might be extremely low, are great for horticulture. Because you put it on small acres and put it on high volume, high cost. But I can't see a corn or wheat farmer taking 500,000 hectares and dumping biochar on it. Anybody work with biochar? A little bit. It's a fine powder, right? Okay. What happens in Kansas, the wind blows, right? It's dust bowl, right? So you have to till it in. I just told you the tillage was a bad thing. Okay, so for large agriculture, our great mollisols around the world that's under agriculture, I just don't think it's cost effective. But I think in degraded land, this is my opinion, I'll probably get criticized for it, but I think I don't think it'd be cost effective. I think I would work on some of the things I talked about here, rotations, deep rooted crops, less tillage, but I would concentrate on more of the degraded soils, ones that are restoration of degraded lands, and purposely apply there. I think you can make a bigger bang for the buck, for the dollar, whatever I forgot the check, krona, is it? Money, whatever the money in check, is, but.
Interchangeable.
Oh, yeah, right. Or cryptocurrency, right? But anyway, I think I think you need to be smart where you apply, but I think biochar has a potential, but I don't think it's a panacea that everything it's going to be, in my opinion.
It's nice, it's nice that you work with long-term fertilizer chain
Yeah,
management experiments. And for example, in the Czech Republic, we have so big problem with the maintenance of such experiments. Because you have projects for 3 years.
yeah.
And then you must look for another money, so it's so problematic. Do you solve it in some way?
Well, this was, when I first arrived at K-State, I got a 3-year federal grant, right? But I did my PhD at Kentucky. So it's on an experiment that started in '69. I was there in 1980, so it was 12 years old or so. And they still got it going today, I think. And so I knew the long-term benefits of no-till systems and long-term experiments. And there's a book out about their long-term experiments globally. I mean, the Rothamsted experiments are the world's longest, and that. So yeah, it was supposed to be 3 years. I decided I wanted to keep it going, and I just cobbled together funding, different research grants I got, but then in between there were some lean years, and it was low maintenance. We had students that go out and put the compost on by hand, and then we farm crew would plant, but we kind of kept it going. Some years would be low, no measurements or a few measurements, but we kept like a 3 or 5 year interval. We were always doing carbon down at depth. We archived those and stored them in the facility. Like I said, administrators, they thought, why are you storing all these soils up? I got a whole room full of soils, right? But yeah, now it's historic. I can't quit it now because it's so valuable. Yeah, that's the problem. We don't keep experiments, and we would have never seen this, right? Nobody's ever seen anything like this because we did keep it going. And like I said, I can't stop it now.
So our faculty is involved in several projects related to decarbonization processes. And my question is, what is the potential of all these plants and crops, in your opinion, in comparison to chemical technologies, converting carbon dioxide into inorganic materials and organic solids, for instance?
Yeah, great question. So we just published, it's a technical paper now, and it was reviewed by the National Academy, but it's not in a quote peer-reviewed journal, but it's a CAS report, and I can get you the reference. We had the potential for U.S. agriculture to be greenhouse gas negative. And in that report, we talk, it's the low-hanging fruit. We can do no-tillage now. We can do crop rotations now. We can do perennialization now, right? And cheaply. We can deploy those technologies now. And for the next 20 or 30 years, we can get some of these things here. The chemical technology, at least when I served on the IPCC, it's, so agriculture technology, we can do it for $10 to $30, well, I guess it'd be less than that per hectare, right? A lot of the chemical technology, they're talking about geologic sequestration, pumping CO2 down into geologic structures. That's over $150 per ton of CO2. And it's really expensive. Now, maybe in 30 years, technology, improvements will make it cheaper. But right now, ag and forestry are the available technologies to save some time for the future. And the more we can do now means there's less burden in trying to reduce CO2 in the atmosphere. So I'm sure there'll be chemical technologies that'll be easier because ag is so diffuse, or soil management, forestry, ag. It's cheap, but it's also harder because you got thousands of land managers, farmers, and trying to get that all coordinated is difficult. But it allows us to get started, to get some of the other technologies, and I think renewables, solar and wind. If you asked me when I served on the FCC 2007, you asked me solar, when it be competitive, I thought 2050. Right now, you can build a solar plant cheaper than a coal-fired power plant, or the cost of electricity is cheaper than solar, and wind is cheaper than wind, or than coal. So what's that been, almost 20 years ago? I would have never thought some of those technologies would be competitive. So we're going in the right direction, but still, we can make ag, at least in this report, we feel that with nitrogen technology, carbon sequestration, some other things, we can make ag carbon negative for ag. So we can make it neutral and also help out with some of the other sectors. Yeah.
One small question, my own question, let's say. You were speaking about native prairie. So what is the percentage, let's say, reminder of native prairie in Texas, Kansas, or US?
Oh, I should know. I know that. I should know that. I do know that number, but I can't recall. It's a couple percent. Most of it. Kansas has the largest tract of uncultivated prairie. You're there, that Flint Hills region, because it's so hilly and rocky, it was never plowed. If it was flat and deeper soils, it would look like Illinois and Iowa all plowed up in corn and soybean. But so it's a few percent. There are some agricultural lands that were vulnerable to erosion, and there was a government program to plant CRP, Conservation Reserve Program, that paid farmers to put it back into perennials. Hopefully they're gonna, who knows, with this current administration, who knows what day it is. But if they keep those programs going, I'm actually working with a group to find out how much carbon is stored in those restored prairies. And then can we incentivize and pay the land managers to keep it in prairie, because once they plow it, we're gonna lose some of that carbon that was put in with perennials. But yeah, it's a small percentage that's still native.
More questions?
Do you need some special crops for local agriculture
For no-till agriculture?
or local agriculture?
Yeah.
To me, like, some new meters or.
Yeah, I mean, well, yeah, I mean, generally there's new cultivars every 2 or 3 years, right? The breeders. It helps, I don't know what your opinion is, but it helps to have genetically modified crops because they're herbicide resistant, and that allows you to no-till. Because then you don't, the reason tillage was done, one, for 2 reasons, at least 2 reasons. One was to get a good seed bed. So they tilled it several times. I'll tell you a story in a minute. But they tilled it several times to get a firm seed bed and allow the planter. Now with the engineers, we have equipment that can get a good seed soil contact without tilling the soil. But then the other reason they tilled was to control weeds. And so with herbicide technology, it allows you to do no-till. Now you can do no-till organically without herbicide, but it is much harder. But I know there are producers, farmers that are doing it in the US and particularly in Brazil. With cover crops, they're what's called crimping. They run the cover crop and then they have a kind of a roller that crimps it, doesn't kill it, but desiccates it without herbicide. And that mulch suppresses the weeds. So you can do it. It's a little riskier. And I've told people, you got to keep everything on the table, all the technologies. But the GMO technology has been what advanced no-till in many places, but now there's ways to do it non-chemically. But I still want the breeders. So in certain places around the world, and I'm probably north of here in Germany and that the soils are wet and cold. So a lot of times they till the soil to dry them out and warm them up. And so they're trying to grow corn in Canada and they're tilling it to warm up the soils. And I'm going, maybe not planting the right crop, right? So we need the breeders to look at crops, seeds that can withstand cold, wet soils for a period of time until the weather warms up. I still want breeders to look at roots, right? To look at deeper roots, different root structures and that. To help? I can't, the window is, I can't see your face, so. Yeah.
I have another question, more related than to our agriculture. So do you think carbon, at least carbon neutral agriculture, productive to feed what we need is possible or not?
No-till for food production.
Yeah.
Yeah. Well, that's the thing is, so in some places, particularly if enough precipitation, you can grow 2 crops in a year. So Brazil, they can actually grow 2.5, 3. We can do 2 because what happens, we can do wheat and then after wheat plant what they call double crop soybeans or corn or sorghum. The reason for that is that if you rely on tillage, you harvest the crop, you till the soil, and then because you got bare soil, it evaporates, the moisture evaporates. So now it's drier. Plus you've lost 2 weeks of time. Because that time to till it and get it prepared. you lost 2 weeks. Now, no-till, because it conserves, I didn't talk about it, conserves moisture, that residue on the surface conserves that moisture. So now I saved 2 weeks, plus I got more moisture. And so I can do 2 crops in a year. So now I can intensify that and diversify that system. So now I can do 2 in Kansas or Southeast United States. I don't know about here, but now I can grow a small grain and then I grow a corn or soybean after that. And Brazil, they can do it easier because they got, 60 inches. So they got, 150 centimeters of rainfall a year. Plus that they don't have the cold temperatures. And so I've seen pictures and I thought it was canned, but it is canned, but they will have the combine and they will have the planter right behind the combine. In sequence. Planting right behind, because they want something green growing in that living roots around year round.
Have still a few time left. Or little time left.
Yeah, the other thing on the no-till is that because you're conserving water, with changing climate, the yields are better in no-till systems than in till systems because you got more water available. So as climate gets riskier or more extreme, no-till is going to be almost essential to maintain food security and profitability.
If nobody, I would have one more
Sure.
question, maybe a little bit more complicated. You have showed several results of the carbon balance based on carbon-13 ratio, and carbon-13, carbon-12. On the other hand, we know that the fossil fuels are more carbon-13 rich, so the carbon-14 is increasing in the atmosphere. Cannot this somehow disturb or systematically make
14 or carbon-13?
13?
Yeah, so because of the photosynthetic pathways, that's the nice thing about using C13. Yeah, C13 is going up a little bit or changing a little bit, but there's a huge difference in C3 and C4 pathway. C4 plants, the carbon discrimination, you get a -12, whereas C3 plants is -22. So if we're talking a small difference, there's still a large gap to enter that. And actually, the soils that we started with in 1992, they are like minus, because it was C4 originally, prairie, but then for 50 years it was C3. So it went from 12 to not to 22, but it's like at 17 or 18. So there's still plenty of sensitivity there to separate out C4 and C3. So the minor differences, well, from a chemist standpoint, they might be significant, but from an ecologist standpoint, those are minor differences that we can still use C13 signatures to look at sourcing of carbon.
So you don't expect systematically false results of, no, let's say false, but systematically biased results because of the higher ratio of fossil.
Yeah.
CO2.
Not, I mean, if you're doing some refined soil chemistry, there is actually during decomposition some discrimination, but in agronomic type studies, it's not enough to affect the interpretation. If you're doing some natural abundance and looking at discrimination by the microbes between C13 and C12, yes, but not on a 10, 30, or maybe, hundreds of years time cycle that can be useful or can affect that. But so it's temporal scale and the quantity that's important. Good question, really good question.
2 minutes left.
Yeah, go ahead.
Are you studying also other organisms like earthworms or other invertebrates and how they influence the structure of the soil?
Sure. Yeah, so earthworms are, well, burrowing organisms, but earthworms are usually a key indicator. Because what they do is they create the pore structure because of burrowing, but also they produce slime or polysaccharides that help, so they can help move through the soil, and they push the soil and they help build structure. So earthworms are important. In this experiment, that, and one of the reviewers said, well, you know, what about burrowing fauna and earthworms? But we would have seen that from day one, not until 15 years later. So they weren't having a major effect in that. Just as a side point, I think it's interesting, at least in the US, the earthworms that people are using as an indicator for good soil health, they're invasive. They came from Europe. They're all over the United States, and actually they're a problem in native forests because they affect the nutrient cycling. But we're using an invasive species as a soil health indicator, which seems to me kind of bizarre. But they're doing good things, well, at least in ag systems, and not so much native, but they're from Europe. They came over when the migration, I don't know, maybe they're German earthworms, I don't know where they came from in Europe.
I don't know either. But for vermicomposting, we use the Brazilian or Argentinian worms on the other hand.
Yeah, okay. But the soil fauna are really important, and certainly in native systems, like, well, even the native prairie, they aren't they're called endogenic earthworms. They don't come to the surface. They're smaller and they stay within the soil profile. The ones that people see that are associated, what they think with good soil, are right now epigenic. They come to the surface to feed, and those are the fishing worms or whatever that people associate with. But even the native prairie doesn't, if they're not invaded, they wouldn't have the big fat earthworms that people associate with. Good, yeah, but they do have effect, generally a positive effect, except they're finding out that they help recycle too fast, and so in native forests they're losing nutrients and affecting the forest ecosystem.
With the invasive earthworms.
Yeah, the ones from Europe, from here.
We are also invasive species. Okay, we are out of the time. Nevertheless, now it's scheduled 20 minutes coffee break. Yeah. The aim of this coffee break is to not left any food waste. Yeah. So eat all what is there, otherwise it will be wasted and it would be undesired. Yeah. And in 3:50 we will be continuing with the students of landscape reclamation.
So, who has the presentation?